So today I'm planning to finish the course. So it may extend to around 1 hour. Okay. So today's class is on applications of the techniques we learned in recombinant DNA technology, basically cloning in the field of agriculture. And as we know that when we talk about RDT and agriculture, then obviously it's the transgenic plants or the genetically modified plants that come into mind. And the need to make these transgenic plants is because they can be resistant. When you want to improve upon the natural population, the wild type population. So the transgenic plants that have been created, they have been in the context of providing resistance to various stresses. It could be salt stress and disease resistant, pest resistant, frost resistant, herbicide resistant, insect resistant. All these transgenic plants have been developed uh, because it was realized that there was a lot of crop damage because of many of these things that I just mentioned. So the idea was that can we make the natural population resistant to these various stresses or the pests that exist in the environment. Then I had also mentioned to you yesterday also that the plants can also be used as a factory for making pharmaceuticals. I told you about the concept of plant farming or animal farming. So yes, many of these they are being considered if we can harvest the various recombinant proteins involved in means the ones which are used for therapeutic purposes, can they be made in the plants. Then also you want to make plants which are healthier or having higher yield as compared to the normal crop, or with the improved shelf life. So various parameters which you think can be improved in plants. They use as factories for making biofuels or various drugs, or just simply the concept that the plants, if they can absorb toxins, then they can be used in the field of bioremediation as well. So it is just the ideas that come in your mind that have to be very innovative. The tools we do have in place, we know that if you want to alter anything in plants, how you can do that, how you can transfer a gene in plant. You need to know which combination of genes need to be altered, which pathway needs to be altered to improve a particular quality. So in today's class, I will be discussing a few examples, few success stories. Obviously, I cannot have a class on transgenic plants without mentioning the BT crops. So first, I'll be telling you about how insect-resistant plants they were developed. Then example of herbicide-resistant plants, the story around it. Then some flavor saver tomato and terminator technology. So these are some things that I'll be covering in today's class. So insect resistant plant, the need came into picture because it was realized that many crops they were damaged because of the various insect infestation. So the idea was that can we make the plants which are resistant to these insects, because in means currently it was realized that yes, you can kill the insects with the help of various insecticides, but then these insecticides, it was realized that they are comparatively non-specific. The problem is that they are highly toxic. Also, they get into the food chain, affect the ecosystem, and also because insecticides, they are sprayed on the plants from above. You must have seen that it is through a particular sprayer that is used. So the insects that were killing the plants or affecting the plants from the under surfaces, they were saved from the effect of these insecticides. So definitely the method that was used to kill the insects, that was not enough, and there was a need to find the ideal insecticide, basically which will be comparatively much more selective in nature and not just kill every insect, maybe a harmless insect also. and also it should be biodegradable. It's not desired that it gets into the ecosystem. And also, if we find a way that all the insects which also affect through the undersurface of the plant, they should also be killed. So then there was a search that how we can do that, and to the rescue came, means I would say that a rescue came from the bacteria for a plant. So there was this bacteria which was discovered, Bacillus thuringiensis, and when the scientists, they were studying the defense system, that how this bacteria uh, defends itself from infestation by various insects. Then they came across a very important protein called delta endotoxin. So these bacteria, during sporulation, it forms intracellular crystalline bodies that contain an insecticidal protein called delta endotoxin. Now, this delta endotoxin, it is highly poisonous to the insect, and it was mentioned that 80,000 times more toxic than the insecticides, and is comparatively selective, which the property that we were looking for a good insecticide, yes, it indeed had that. Okay, so how this? So this delta endotoxin, it is produced by the bacteria, and when the insect it digests, I mean it eats the bacteria, then this endotoxin, it acts on its gut, and sorry, so it gets into the stomach, and at high pH it gets activated. So once we have an active toxin, so it creates holes in the gut of the insect, and then the insect dies within few days. So definitely it was realized that if we have this delta endotoxin, then the insects may get killed. So with this observation, there was this idea that can we transfer this delta endotoxin to the plants itself, so that plants are themselves making this particular protein, and when the insect digests or infects the plant, then it will die because of the presence of delta endotoxin. So how do you transfer this this bacterial protein to the plants? So it is simply by cloning. You amplify this particular protein, this gene, and then you introduce it into the plants. But then the journey was not that simple. So it was. So let me tell you about delta endotoxin. It's around more than 1,000 amino acids, but it was realized that it is only within around 600 amino acids that you have the toxin activity. So it was realized that there is no need to clone the whole gene, but even just the N-terminal portion, if we clone it, then it is good enough. So then, can we just take this portion of the bacterial gene and transfer it to plants and assume that a good protein will be made? No, that was not the case, because you have to realize that it is bacterial origin, and we are transferring it into plants, which is eukaryotic. So there is a thing that the problem of codon bias was there. So it was realized that we have to alter the amino acids in the original bacterial gene to make it into means make it effectively translated when transferred into plants. So then rather than cloning and doing single mutation at particular codon positions, they actually artificially synthesize the initial N-terminal portion of this particular protein. So where the preferred codons, which were in this case, let's say I'm talking about maize, when they made BT maize, these preferred codons, they were incorporated, and we had a artificial gene of around 600 amino acids. Then it was realized that obviously we cannot have the promoter which is endogenous to this particular gene in the bacteria, because the bacterial promoter will not be recognized by the RNA polymerase machinery within the plants. So this gene, it was cloned downstream of a promoter which is active in plants. And I told you that CAMB, this Colimo mosaic virus promoter, is generally used. CAMB is one of the very commonly used promoter in plants, and this gene was cloned downstream of it. Then obviously polyadenylation signal was also added on the other end, and this cassette then had to be transferred to maize. Now, if you remember, I told you that in dicots, it is you generally go for agrobacterium, while for monocots, agrobacterium technology, at least that time, it was not very effective. It is only now they have made some changes in agrobacterium to make it effectively transform the monocots also, but that time it was not common. So in that context, I told you there are various different methods by which you can do monocot plant transformation, one of which is bombardment. So what they did is that they took maize embryos, they bombarded this cassette, the cassette that they had made, which was coated, DNA coated microprojectiles. And these, they bombarded a number of embryos. So a number of transgenic plants, they were developed from it. And how do you develop a plant from an embryo? By, by giving them proper conditions and also altering the concentration of the various hormones that I told you. So finally, we got various transgenic plants, and then these transgenic plants, they have to be screened. Why they have to be screened? Because there is a possibility that when you bombard or when you introduce your DNA cassette into plants, then that particular cassette, it may get incorporated at various places uh, in the whole genome. So in one transgenic plant that you have made, it may get incorporated in some euchromatic region, while in some other it may get incorporated into the heterochromatic region, and so on. And depending on where it gets integrated, how actively transcribed region that is, will give rise to various transgenics with varying levels of protein overexpression. So then you have to screen your transgenic plants. And once you get your transgenic plant, then you test it that, okay, I made a BT maize, which is overexpressing this delta endotoxin, but how effective it is to contain the this infestation by insects. So you do these field, or in controlled condition, you do these experiments where you take wild type plants and you take your transgenic plants, and you put the insects on them. So it was realized that the maize plant, the transgenic plants, in that, when they monitored that how much damage has been done by the insects, it was found that in transgenic plants, the tunnel was only 6.3 centimeter, which was made by these insects, while for the white type, it was 40.7 centimeter. So it was like a huge difference in the damage which was created by the insects, and that was the success story of making the first BT maize. And why maize was specifically chosen to make this BT, BT maize was the first crop to be made, was because of the fact that maize was the crop which was not very well served by the conventional insecticide. Why? Because the pest which infects this maize is this European corn borer, which tunnels into the plant from eggs laid on the under surface of the leaves. So you can understand, because this insect is going from the lower surface of the leaf, so obviously however much insecticide you add from above, it won't be very effectively acting upon the lower surface. So that's why maize was considered as a group crop to check the impact of this transgenic process. Now, another aspect came, and that is chloroplast transformation. And why this need was there? Because let's say when you make this BT maize, there is a chance that this transgenic plant, the foreign gene that you have introduced into it, it may be, transferred to the other plants which are present in the environment by cross pollination, etc. So there has to be some way. One way is obviously that you grow it into in a completely controlled environment, but then another aspect is that can be, completely block the transfer of this particular gene to the other plants which are present in the environment. So then the idea came that how about we clone this particular gene not in the nuclear genome but in the chloroplast genome. So what will happen that, let's say, if your gene was cloned in the nuclear genome, then the pollens which are made, they will also have the nuclear genome, and the gene, the foreign gene, there is a chance that it can be transferred in, or spread in the environment. But if I clone my gene in the chloroplast, then because the pollens do not have the chloroplast, then that particular option is completely gone. So that way I can ensure that my gene will not spread to other plants in the environment because the pollen does not have the foreign gene. So this was one aspect, and another aspect was that I told you that if you clone your gene in chloroplast as compared to you in nucleus, then because there are multiple copies of chloroplast in a plant cell, so you will have, more copies of a particular gene that you want it to be expressed. So another aspect, so if I ask you that what are the advantages of cloning in chloroplast, one would be the escape the of the foreign gene, it is restricted. Second is you can get more amount of a transgenic gene, and third is transgene, and third is that there is, because chloroplast genome is bacterial in nature, that you must be knowing, both mitochondrial and chloroplast genome, they are more prokaryotic in nature. So I do have an option that in case I want to express three genes at the same time, then I can express them in the form of a polycystron. Because in prokaryotes, there is an option that polycystronic transcripts are there, genes are there, where two, three, or five different genes, they are under control of a single promoter, and all of these are very efficiently expressed. So let's say if I have to transfer three genes in nuclear genome, then for all the three genes, I have to do separate cassettes for each. Each has to have its own promoter. So that's more difficult as compared to if I take one promoter and three genes together. So they tried this particular option also. So this is for so these delta endotoxin genes, they are also called as cry genes because these are crystal proteins. If you remember, I told you just few minutes back. So that stands for cry proteins, and this is the cryoperon. So here, what they did was that not only they expressed this delta endotoxin gene, but also along with it, they expressed two other proteins as part of the polycystron, which helped in its overexpression and folding activity. Means it was much efficiently folded as well as expressed when these helper proteins were there. And what they found, that they had a huge amount of toxin made by these plant cells in which chloroplast transformation was done, where over 45% of the total soluble protein in tobacco was this cry protein. So this was like a huge achievement. And when they compared this transgenic plant, in this case, it is BT tobacco that they had made, as compared to the wild type, then they found a huge difference. You can read all this, that when they compared, the larvae were dead in the transgenic plants, while there was a lot of damage in the wild type plants. But at the same time, I also want to tell you that the chloroplast transformation is not a easy thing. It has been successfully done in only a few plants till now. One of them, the early ones being tobacco, but it is considered a very difficult process, and scientists are still trying to optimize this particular method. Now, the point is that okay, you make a insect resistant um, crop, and by incorporating a bacterial gene in it, but at the same time, there is a possibility that whatever resistant plant you are making, let's say you're making a virus resistant plant, or you're making a herbicide resistant plant, or you're making a insect resistant plant, then there is a chance that even insect finds a way to become resistant to the toxin that you are introducing in the crop. We know it's a very dynamic, nature and evolution. And as a result, there is a high chance that insect also over a due course of time become resistant to whatever toxin we are given. So then, what counteracting strategy scientists have used or developed to counteract the insect resistance? I'll just come to that. So one method they found was that how about that instead of one type of toxin, you make a transgenic plant that makes two different types of toxins. Okay, so I told you that as compared to the insecticides that are used in the field, which are highly non-specific in nature, the cry proteins, these delta endotoxins, they are comparatively specific. So different strains of Bacillus thuringiensis, they make different cry proteins. Some makes cry 1, some makes cry 2a, cry 2b, etc., etc. So all of these have comparatively different sensitivities towards different types of insect. So as a result, there is a possibility that if I introduce two different cry proteins in this particular plant, then maybe the insect survives or becomes resistant to one toxin, but at least it will die by the other toxin. So this was the idea behind it, and was found to be successful also, at least for some transgenics that they had made. Another possibility is that how about, see, any organism, it develops resistance when they are exposed to a particular chemical or a toxin for a prolonged period of time, and over time they develop these resistance mechanisms. So one option is that how about we have a temporal control over the toxin production. So in means we only expose the insects to only a short period of time to this toxin, that they're not totally unprepared for it, and they die when they are exposed to this toxin, and they don't get time to develop any resistance. So in this case, what they did was that they made the transgenic plants with carrying this delta endotoxin under control of a promoter, which only becomes active when it is at the cob stage and only within the cobs. So, or I think it was a cob stage. So the idea was that when the plant is growing, generally insects, they don't, affect it. It is only at this cob stage that the insects, they get attracted to this particular plant and damage the cobs. So the idea was that can we have a controlled expression of this toxin, and which only gets expressed in the cobs during this stage, and the insects are totally unprepared for it, and then they die. So this was also a very good strategy. And the third strategy that scientists, they thought can be effective is by the method of mixed planting. That is, that in a field where these transgenic plants are growing, how about you also grow these sensitive plants, these wild type plants, and with the idea that there will be insects which will be uh, which will be feeding on these transgenic plants, and there'll be insects which will be feeding on these sensitive plants. So at a time in a particular field, we will have the insect population. We will have both sensitive insect population, because there will be many insects which will be affecting these, and there'll be some insects which will be exposed to these transgenic plants and may, develop resistance over time. So at a time in a field, we may have a sensitive insect population, or the normal insect population, and the resistant ones. Now when the sensitive and the resistant population they mate, and the progeny that is made, it was realized that actually the resistant trait is recessive in nature. So the progeny insects that will be made, they will be again sensitive in nature, and they will die. So this is another method of planting that they came up with, where they don't have to, think about putting two toxins together or having a temporal control, but just by, planting strategies, you can ensure that we don't get an insect population which is completely resistant and totally not getting affected by the transgenic plant. So yeah, so this is all about the insect resistant plants that were developed. A huge success. I've given you the examples of BT maize and maybe BT tobacco, etc. But then there is a whole battery of BT crops that have been made, because it was realized that so many different crops, they are affected by the insects, and there's a need to develop insect resistant crops. So we have in the market BT potato, BT brinjal, BT tomato, BT cotton, and tobacco, and so many different BT soya bean, etc. Okay. Now coming to another example, which is herbicide resistant plants. So what is herbicide? Something that kills, a chemical that kills herbs. So why do we need herbicide resistant plants? Is because in when you grow plants, your desired plants in a field, you also have the growth of these weeds in the field. So these weeds, they are generally herbs, and to kill these weeds, farmers, they have to very carefully spray these herbicides, because these herbicides, they in general kill any plant cell, be it for a weed or be it for a crop. So the only way to ensure that your crop is saved and only the weeds die within the field is by spraying them properly only on the weeds. So it becomes very difficult. And then there was a need to find a way that if there is a strategy that the plants are totally resistant to the herbicide while the weeds only die, and the farmers don't have to be very careful while spraying, and they can just spray herbicide over the whole field, and the plants become resistant, means the crops survive while weeds die. So let's understand first that what is the mechanism of action of the herbicide I'm talking about. So the herbicide that I'm talking about in this case is glyphosate. Okay. So first let me tell you this particular pathway, and then I tell you that how glyphosate it affects it. So shikimate is converted to this particular compound, enol pyruvyl shikimate phosphate, by the action of an enzyme called EPSPS. So EPSP stands for enol, P for pyruvyl, and then shikimate, and then phosphate, and the last S comes from synthase. So EPSPS. So this enzymes convert shikimate to this, and these are then converted to three very important amino acids: tryptophan, tyrosine, and phenylalanine. And if this pathway is perturbed, then you will not have the synthesis of these particular amino acids, and the plant cell or any cell, it will die. So it was realized that can we, kill this pathway. So that's how this particular herbicide was developed. So what herbicide does is that glyphosate, it competes with another compound called phosphoenol pyruvate. And what it does it, so EPSPS, only when it is bound to PEP, it becomes active and then goes for this pathway. However, if we somehow inhibit the activity of EPSPS, uh, in this case, what glyphosate does is it competes with PEP. So glyphosate binds with EPSPS, and as a result, it is inactive, and we do not get these amino acids being made, and as a result, the beet dies or the plant cell dies. So the idea was that, okay, if this is the method, then how about that I make transgenic plants which have very high levels of EPSPS. That is, that even though glyphosate is trying to inhibit EPSPS activity, but because we have very high level of this particular enzyme, so plants will be comparatively resistant to it as compared to the weed. Okay, so the plants will survive while weeds will die. So yes, the initial attempts to make a herbicide resistant crop was by overexpression of EPSPS, but it was realized that yes, it was giving some resistance, but it was not very apt. I mean, it was not enough for the plants to completely survive as compared to the weeds. Then another idea came, that how about we engineer in the plants the EPSPS enzyme, which is resistant to the action by glyphosate. So then a big search, it happened among various plants and bacteria and so on, that can we get a enzyme which is a mutant enzyme, or let's say altered enzyme which is glyphosate resistant. And finally they came across enzyme from a bacteria. You imagine that how much search is done, and what ideas these scientists come up with. So they came across this agrobacterium strain, and the EPSPS gene from this strain was found, or chosen because of its, not only it had very high catalytic activity, but also it was highly resistant to the herbicide. So then the idea was that can we engineer this particular protein into the plants. So then it was realized that since EPSPS is finally located in the plant chloroplast, so the agrobacterium origin, this gene, it had to be cloned in chloroplast. So, or sorry, it had to be targeted into the chloroplast. So what they did was that they cloned this gene, and they introduced that into the plant using obviously agrobacterium, and only thing was using the TI vector. And the only thing is that they made a fusion protein, and in which on the N terminal end they added a leader sequence, which when the protein will be synthesized will act as a leader sequence and will direct the enzyme across the chloroplast membrane and into the organelle. And okay, so in this case, they did not use agrobacterium mediated transformation, but rather they have used biolistics. So it depends on that in which crop you are using and which method of transfer you are using. You very well know that there are multiple methods of transferring a particular gene into the plants. Okay, and after regeneration, when they get the transgenic plant, it was found to have threefold increased herbicide resistance as compared to the control, which was good enough. So these were some of the early, Roundup Ready was the name given to these herbicide resistant crop, more specifically in this case, glyphosate resistant crop, and it was developed by Monsanto. Then the idea was that, okay, in this case what you're doing is that you are making in a way this pathway resistant to the effect of glyphosate by adding a mutant or altered enzyme. But finally glyphosate was found to accumulate in the plants, and then it was affecting the growth of the plants or reproduction of the plants. So then the idea was that, is there some way that we can inactivate glyphosate when it comes in case of the crop plant, while weed plants which are not transgenic, they will die. So then the idea was that can we inactivate or detoxify the glyphosate in transgenic plants? You think about the various strategies that scientists come about with. So then they came across an enzyme called GAT, which is glyphosate N-acetyl transferase, uh, which brings about detoxification of glyphosate by converting, or let's say adding a acetyl group to it. Interesting. So then the idea was that from which source do we get the GAT enzyme? So they first used the this Bacillus licheniformis enzyme, sorry, bacteria, which has this GAT enzyme. So they took GAT enzyme from here, and then they engineered it and made a transgenic crop. But then it was realized that even though this enzyme is very good in detoxification process, but the rates are very low. So then the genetic engineers, they try to find that how can I, change the activity of GAT so that it becomes much more efficient in nature. So I've told you that, not only in agriculture, but in many different scenarios, you have to make a better enzyme. And it is for this reason I had introduced you to, maybe two classes back, I had introduced you to various methods of mutagenesis. So one method is you just go for random mutagenesis, and there is a chance that the altered or mutant enzyme that you get is much more effective as compared to the wild type enzyme, right? One method is that, or you means very meticulously look at various regions in the GAT enzyme and try to find the regions which may impact the activity of GAT, and then you go for site directed mutagenesis. So if you remember, I told you about directed evolution or the other method, etc. So you need to go with all these strategies, but in this case they went for multi gene shuffling. You remember domain shuffling, I told you. So this is an excellent example of it. So it was realized that this bacteria has three native GAT genes. Okay, I told you that many genes, they are present as a multi-gene family. So there were three native genes, and then they did those domain swapping experiments. If you remember, cutting the gene by enzymes and putting the various domains next to each other in various permutation and combination. And so in they did one round of shuffling. They got one final mutated GAT, which was much better than the previous one. But then they again took that as the starting point, again did multi-gene shuffling, and again did multi-gene shuffling. So they did 11 rounds of multi-gene shuffling, and the new GAT gene that they found, that they made, actually it was found to be 10,000 times more active than the previous GAT. So do you see, this is a major achievement. I'm just saying that the techniques you already know. How do you put it across for solving a particular problem is means your brain has to be very active, that which gene has to be used and how it has to be altered and which particular problem it can solve, etc. So yes, so this is the story about development of a herbicide resistant plant. And I've told you the examples of how insect resistant plant was made or herbicide resistant plant was made. But then obviously it's not restricted to that. There are so many different examples in which transgenic plants have been made. Some are insect resistant, fungal resistant, bacterial resistant, virus resistant, herbicide resistant. Some will have better yield, some will have better content, will have altered fruit ripening. I'll just come to this example, modified fat, and so on. And what I want to bring to your attention is the fact that the genes have been taken from various different sources. It is how smartly and how observant you are, that you can come up with the transgenic plant with the excellent character that you seek for. So in some cases, the source organism of the transgene has been another plant. In some case it is bacteria, in some case it is virus, and so on. So this means you have to appreciate the fact that, see, in this case a rat is the organism. So it is not that you're working on plant field, you only have to study literature only related to plant field only. You should also be aware what's going on in other different organisms, what research is going on. And that's why I always tell you that you should attend all the seminars, irrespective of which field you are working in your respective PhD or MSR labs. Anyway, so the two examples I gave you were the examples of gene addition. But then there is a option, there are many cases in which you would like to inhibit the expression of a particular gene. And in that case, you will have to go for gene inhibition approaches or um, inhibition approaches, or sometimes it's also called as subtraction, gene subtraction. So anyway, so the very good example, or the first example of the plant, transgenic plant that was made, is the flavor saver tomato, in which the gene inhibition approach was used. So the problem that was faced by the farmers was, regarding these tomatoes, were that when they would harvest the tomato and take it actually to the market and ship them and all those things, by the time it reaches the market and for actual consumption by the people, they would get spoiled. So during transportation, many of the tomato that they have harvested, they would get spoiled because of the ripening process. So then one option these farmers had was that, how about you just pluck the tomato when they were green in color, means they were very much at an early stage, and then take to the market. But then that was not a good option, because then the flavor of the tomato was lost. So tomato has to be harvested at a particular stage only when you have a perfect flavor. And they had to find a way that even when they pluck the tomato at this particular stage, it the ripening process is not fast, and they are able to take it to the market without affecting its quality, etc., and the fruit stays firm and is not damaged. So then the idea was that, how do we identify the genes which are involved in the ripening process? So there comes the role of basic scientists who identify various genes and pathways involved in a particular process. And one of the genes that they came across was polygalacturonase. So this particular enzyme, it acts on pectin and makes the this fruit coat. It acts on the fruit coat, this the cover of the tomato, and then damages it. So one way was that, how about we decrease the level of this particular gene, and then get a transgenic plant which has low levels of this, so the fruit will not be spoiled. So, and we can also harvest it at the right stage. And indeed, that was the case, that when they inhibited the level of this particular enzyme, then the plants were much more resistant to spoilage as compared to the control plants. Now, how did they decrease the level of this? I've told you, if you have to decrease the level of a particular gene, you can go for antisense therapy, or siRNA based strategy, or microRNA based strategy. So they went for antisense strategy, which is that, let's say you have the gene, the promoter, which gives rise to this particular gene. So they added the gene, the same gene, but in opposite orientation, under the control of the promoter. So what will happen, that the wild type gene will give rise to the sense transcript, and the transgene, which is cloned in the opposite orientation, will give rise to the anti-sense transcript. And because these two transcripts, they are complementary to each other, they will make a double-stranded RNA, because they're completely complementary. And this RNA will be acted upon by dicer, and then the the transcript will be acted upon by RNases, by RISC complex, and finally we see the downregulation of the wild type gene. So this is the strategy that they used in this case, and we saw very good effect on the tomato quality. And these were the saver tomatoes that were developed by a company called Calgene. So however, Calgene, it closed after some time. I think there was some marketing strategy problem or something like that. And then other methods of this tomato the controlled ripening in tomato, they were developed. And one of the other way by which you can control the fruit ripening, it was realized that how about we inhibit the production of this particular hormone called ethylene. So it's a gas, but it also acts as a hormone, and which is very important for the ripening process. So it was realized that how about we decrease the level of ethylene. So how can we decrease the level of ethylene? For that, we need to know the pathway by which ethylene is produced. So the penultimate step in ethylene synthesis is conversion of this SAM to ACC, which is the immediate precursor for ethylene. So then they made the transgenic plants in which the ACC synthesis was inhibited. So in fact, I'll tell you the various strategies that were used for ethylene inhibition. And this is some updated material that I got. Just to introduce you to the fact that how many different strategies are used to make a transgenic plant with a desired character. So in this case, the desired character is that somehow we have to decrease the expression of ethylene. So I tell you, see, these two companies, Agritope and Monsanto, what they did was that they delayed the ripening by preventing the production of ethylene that I just told you. So what they did was that they inserted at least the DNAP, this Agritope and Monsanto, they inserted truncated version of ACC synthase into the tomato that interfered with the endogenous ACC synthase. Then Monsanto, what did they do? That they rather engineered another enzyme, ACC deaminase, and the source was the soil bacteria. So that the introduction of this enzyme, it lowered the ethylene levels by breaking down ACC. So this is completely different strategy. You can use siRNA against ACC. That can be another third strategy. What did Agritope do? So in this case, what they have done is that they took a gene from a phage. So in this case, it's a T3 bacteriophage, and which has this gene called SAMase, that is S-adenoside methionine hydrolase. So if you remember that for ACC synthesis, SAM is also important. So this reduced the level of SAM, which is a precursor to ACC. And in India, another two scientists, what they have done is that they silence two genes which are N glycoprotein modifying enzymes, manosidase and whatever. So what I just want to tell you through these examples is that the ultimate aim is same. You want to decrease the ripening and prevent the spoilage, but so many different strategies may be used to achieve the final product. Which of these strategy will be the best strategy? You have to do various experiments for that and then come up with that, okay, this is a better strategy as compared to the other, and so on. So so these are the ways by which various transgenic plants have been made. In the note section, I have also given you example, how blue rose was developed. So in nature, we never find a blue rose, and then there were scientists who were very, passionate about developing a blue rose. And just so it's a Wikipedia page, just go through. It's a small para that what all methods they used to develop a blue rose. पहले उन्होंने blue color का एक gene डाला. But then it was realized that because the original rose was red in color, you don't get the blue color. So then they inhibited two different genes to remove the original color of the rose, and then only come up with a rose that will have at least some blue tinge. So they have not yet been able to get a completely blue rose, but they have at least achieved till the mauve color. So just read that strategy just to appreciate the fact that to get a particular phenotype, what all different strategies are used. Okay, so these are gene subtraction or gene inhibition approaches. You can understand, you need to know which gene expression has to be downregulated to get a particular character, and so many to control the fat synthesis, or modify a color of a particular plant, or control the ripening process or discoloration or starch content, etc. Different genes have been targeted by different approaches, and to achieve this particular character. So these are some beautiful, figures just to show you that the difference between the wild type. You can see that this has been affected by a particular insect, and these are the insect resistant cobs. The difference between the two, in this case, they have developed transgenic tomato, which is resistant to virus infection. So obviously there are many plants which are affected by viruses. The infection is very prevalent in certain crops, etc. So they developed these transgenic tomato, and these are the wild type. And when they compared the two, and virus was infected in both of these, they found that they grow very well while they don't grow at all. In this case, they have developed a salt tolerant brassica variety, and you can see this is a wild type plant which, when grow at very high concentration of salt, cannot grow almost at all, means hardly, its growth is highly inhibited. But these are the various transgenic overexpressors that have been made by engineering different genes, which have different resistance levels as compared to the wild type. It could be that in this particular figure, all the three transgenics, the same gene was engineered in this. They may all have the same amount of the transgene, I mean, one copy of the transgene, but it could be that in one case the gene got incorporated at the euchromatic region, so you have very high expression, while in some case it maybe has got in some heterochromatic region, you don't see that much expression of that particular gene, so you get a comparatively lower phenotype. So yeah, these are certain example of golden rice being developed, and you can see the difference between the control and the golden rice. So because beta carotene gene has been engineered here, so see, golden rice has been developed with increased vitamin A production through the increased production of carotenoids. So that's why you see this color in the golden rice, and that's why it's actually also called as golden rice. And the idea was that how about we make a transgenic plant which has a higher amount of iron accumulation, and which may serve the problem of iron deficiency in various children, that is children or any individual that is generally seen. So are you fine with 15 minutes more class, because it is this technology I have to tell you, or do you want to go on for the next class on second?
Ma'am, I have a class at 10 and I have
Okay.
presentation on in that.
Okay. So then we will meet on second at 9 a.m.
Okay, ma'am.
Okay. Okay, then take care.
Hello, ma'am.
But yeah, if you have questions for today's class, you may please ask.
Ma'am,
Yeah.
ma'am, what will be the syllabus for the practical quiz? Will it cover only the practical classes or both the classes that we have taken or and the TS?
Both the classes means, I mean,
Means
the class.
the syllabus, ma'am. Will it cover only the classes which have taken by the TS or
Yeah, the practicals that have been taken by the TAs, not what I am teaching in the class. But obviously, they both are associated. So, I mean, it will be basically the practicals that have been covered by the TAs.
okay, ma'am. Okay, ma'am.
For clarification, I can also pass on the email to various students who might have this doubt.
Okay, ma'am. Thank you, ma'am.
Okay. Anything else? Okay. If there is no more question, then have a good day. See you on second then. Bye bye.
Thank you, ma'am.
Yeah,
Thank you,
take
ma'am.
care. Bye bye.
Thank you,
Bye.
Thank
ma'am.
you, ma'am.
Hi, ma'am.
हाँ बोलो. अच्छा, you are